Hello, my name is Francisco Corzana. I am professor at the University of La Rioja, and I am very happy to describe briefly our research project. As you know, COVID-19 is one of the biggest health crises in history. Thanks to science and technology, we have developed vaccines that are effective against this virus. So major vaccination efforts are currently underway to immunize people around the world. However, we know that new variants are emerging, and that's why we consider it's very important to develop a new type of vaccines. So in this context, the main goal of our research project is the design of new vaccines against COVID-19 based on artificial antigens. We will replace several amino acids or peptide fragments present in proteins of this virus, for example, spike protein, with a natural amino acid. In this way, we will obtain derivatives that are more stable against enzymatic degradation and at the same time and hopefully more immunogenic. And this property is very important in order to develop good vaccine candidates. To achieve this ambitious goal, we will follow a multidisciplinary approach based on synthetic chemistry to prepare the peptides and also glycopeptides. These molecules will be conjugated to nanomaterials in order to boost the immune system, so we will use also nanotechnology. And finally, we will perform biological assays in order to check the efficacy of our vaccines in mice. So these are the main lines of our research project. Thank you very much for your attention, and please contact me if you have any question about the project. Thanks.
Hello, my name is Ana González. I am the coordinator of the project's Plasma and Machine Learning group at the University of La Rioja. Just let me introduce you my colleague Fernando Alba, who is going to talk about our cold plasma research line.
Cold plasma has a lot of application in agriculture, food industry, and cancer treatment. We use direct plasma to sanitize oak wine barrels. In this case, we obtained the complete removal of Brettanomyces from wood. As I told you, the energy of plasma could be transferred to water to create plasma-activated water. There is a lot of application of plasma-activated water in food industry and also a great number of methods to create plasma-activated water. Our research group have developed several ways to create plasma-activated water. The difference between these generation methods are related in which way we put in contact plasma with water. Cold plasma could be used to apply functional coatings over heat-sensitive surfaces. In this case, we have applied a two-layer coating over stainless steel in order to reduce the biofilm produced by Listeria monocytogenes. Well, why is this research line important? Well, cold plasma is cheap, environmental, sustainable without using chemical and generating waste. It can be used with heat-sensitive surfaces like human skin, medical devices, food, and surfaces in contact with food. Which issues are we trying to improve? Well, we need to understand the decontamination mechanism, to transfer this technology to the industry, to promote regulation for safety use. For that, our research group participates in two cost actions of the European Union. Thank you very much.
I am César Domínguez from the University of La Rioja. I am going to explain our research line entitled Biomedical Image Processing. This research line consists in the application of mathematical and computational methods to build tools for biomedical imaging processing. The methods that we focus on came from artificial intelligence and are machine learning and deep learning models. This research line is made in collaboration with specialists in life science fields such as biology and medicine. The final purpose of this research line is to develop algorithms and tools that can be used by these specialists in their daily routines. The supervisor research groups is named Computer Science Group from the University of La Rioja. This is a quite big group that has several lines related with the previous one. The co-supervisors are Yolanda Sanz from the Molecular Microbiology Group at the Fundación Rioja Salud. She is working on the study of bacterial resistance. And Manuel Doblaré from the Institute for Engineering Research of Aragon at the University of Zaragoza, and he's working among other lines in cell and cancer modeling. Some examples of the success of our research with these groups are GelJ and SpheroJ. GelJ is a tool that is able to analyze DNA fingerprint images of bacterial, and SpheroJ is a tool that allows the study of spheroids that are cancer and tumor models. Thank you for your attention.
Hi everyone, thank you for your interest in our research. The Organic Photochemistry Research Group in Universidad de La Rioja is interested in the interaction between light and matter in three main research lines: photoprotection, photoswitches, and energy storage. We try to go all the way from the basic research to the final application. So, for instance, we have several patents and contracts with companies. This project is focused mainly on energy storage, a research line in which we have been quite active in the last years, and now we are part of a quite ambitious European project. The idea is to use the so-called molecular solar thermal systems, which are based on a photochemical reaction in which we use the sunlight to turn a parent molecule into a high energy isomer. And in doing so, we can store part of the solar energy into chemical energy. This stored energy can be released through a catalytic back reaction when needed. Using these very basic design principles, a working device was built with excellent performance regarding the heat release. Within the European project, we are now trying to build or design and then build a new device, trying to take advantage of both light and heat coming from the sun. Our participation in this project is mainly focused on the catalytic heat release device. This is a truly multidisciplinary topic in which we use organic synthesis, photochemistry, catalysis, and computational chemistry. So anyone with a background in one or several of these fields would be welcome. We have contact with many different research groups and companies worldwide, so we can offer a truly exciting project and experience. Thank you.
Good morning. I would like to introduce our research group on antimicrobial resistance from the One Health perspective. We are a multidisciplinary group of researchers in different fields as pharmacy, veterinary, medicine, biology, environmental science, chemistry, biochemistry, and food technology, and we work in the Faculty of Science and Technology of the University of La Rioja. We know that antibiotics are one of the most relevant discoveries in medicine during the 20th century, but antimicrobial resistance is increasing at alarming level, compromising the treatment of infection. For this reason, this is the main topic of our research, analyzing the implication of this problem in human and animal medicine, food security, and environmental health, with the idea of taking in the approach of the One Health. In this sense, we analyze commensal and pathogenic bacteria, both in healthy and diseased human, in different types of animal, in food and the environment, with the idea of determining the mechanism of resistance in bacteria of different ecosystems, the way they transfer the resistant gene in the way of plasmid, integron, transposon. We are very interested in molecular epidemiology using the tools of the genomic and proteomic, new diagnostic tool, and also to look for new antimicrobial and new alternative to antibiotic of interest in medicine, veterinary, and food production. Our main topic also is to analyze the evolution of antimicrobial resistance, doing antimicrobial resistance modeling, looking for the relation of antimicrobial resistance with sustainable development and antibiotic footprint. These are the main topic of our research, and here are the link that can be used if somebody is interested in our research. Thank you.
We are going to introduce to you the health economics group of our University of La Rioja that belongs to the campus Iberus. Our research group that is named EcoSalud was created about 25 years ago, and we also collaborate with other scientific forums and professional bodies such as the Spanish Health Economic Association, and also we have international networks for collaboration. We have participated in 7 research projects funded by the European Union in the last years. So the main topics of our research line are the economic evaluation of health technologies. We study also the efficiency of health organizations. We also analyze from a theoretical point of view decision-making models for health policy and management. And we also emphasize the studies of the economics of drugs. That is a quite complex sector. Why do we believe that this research line is important? Well, because the healthcare organization is quite complex and challenging given the growing population needs and also the increasing demand for public funds to pay for the resources. We also consider that the analysis of the efficiency of decisions becomes a crucial issue to guarantee the optimization of scarce resources in context of growing needs. What are we trying to solve with this type of research we are doing? So the decision-making process of the healthcare system in context of uncertainty and budget pressure, and also the management of drugs based on the information generated by economic evaluation studies. Many thanks for your attention.
Hello, my name is Ignacio Matias. I'm professor of the University of Navarra in Spain. The idea of this the proposal is to develop a unique photonic sensing technology for the detection of biotargets of different sizes. The sizes are from the erratic organic compounds from 1 to 20 nanometers, viruses, bacteria, fungi, and so on. We also have the collaboration of Natech Innovation, whose research leader is part of this team. We are going to use also 2 different technologies, Lucimo resonances and Sulfate primer resonances, and also 2 different types of substrate, planar and optical fiber. Here you can see some sensing structure possibilities. We have already studied that is just using holes, strips, or whatever. And also on the right side, you can see a potential application that to detect that it consists of detecting biomarkers in the breath. Depending on this biomarker, you can get some of the diseases like you can see here and here. We have already worked on this in advance, and for example, on the left, you can see here a Lucimo resonance with 2 different peaks to detect 2 different targets, and also we have already worked on the mid-infrared here on the right. You can see. And also in parasubstrate, we have already worked on that dual sensor with silver and ITO for detecting Sulfate primer resonance and Lucimo resonance, and to detect 2 different targets. And finally, also we have already worked on intertitanic photonic sensor like this one, that depending on the number of the strips, you can detect as many targets as strips here. So the idea is very easy, just to also evaluate sensor of underwear guide to detect this peak, and just to fabricate a medical device that can be handled in the palm of the hand. I encourage you to apply for this project and to ResearchLines and become part of our group. Thank you.
Hi everyone. Our research proposal is entitled Evolution and Adaptation of Two-Component Signal Transduction System in Staphylococcus aureus. Our group has been studying the molecular mechanisms underlying bacteria biofilm formation process with the final aim of finding solutions for the treatment of infections associated to medical implants. During these studies, we have been analyzing the family of two-component systems because they are the main sensory system in bacteria and therefore responsible for the switching between planktonic and sessile lifestyles. A canonical two-component system consists of a histidine kinase that in the presence of the right signal autophosphorylates in a histidine residue. Then the phosphoryl group is transferred to an aspartic residue of the partner response regulator. This response regulator, once phosphorylated, is responsible for modifying the expression of a set of targets, and with that, it couples the cellular physiology with the environmental condition. During evolution, each bacteria has gathered a collection of two-component systems, each of them responsible for sensing and responding to a different environmental signal. Thus, the number of two-component systems in a particular bacteria is proportional to the number of environments in which this bacteria is able to grow. In a previous study, we sequentially removed the 50 non-essential two-component systems in a clinical strain of Staphylococcus aureus. The resulting strain contains a single two-component system. In this project, we will use this powerful and unique genetic resource to identify the changes that Staphylococcus aureus needs to undergo in order to gain the capacity to sense and adapt to a new environmental condition. We hope that this proposal is of interest to you. Thank you.
Hello, I would like to discuss a little bit our research group and the main lines of research that we are currently undertaking. So we are a group composed from people from the University of Zaragoza, the Hospital Servet and the ITA, and we focus on the development of new methodologies for trace elemental and isotopic analysis. We have many years of expertise with a number of atomic techniques based mostly on atomic absorption or ICP-MS. And we have a current focus on biomedical application and on nanoanalysis. I don't have the time to discuss everything that we do, so I will focus on one particular example. It's minimally invasive analysis using what is called as dry blood spots. This is related with blood sampling. We all know what venipuncture looks like and advantages and disadvantages. But alternative has been explored for many years, and perhaps the most successful one consists of depositing a few droplets of blood onto filter paper. This is very interesting, not only for children, but also because this material is quite stable. You don't need refrigerators, and also enables to develop home collection schemes. And this is something we are particularly interested. We believe that this scheme of preparing your own sample and sending to the lab could be appropriate in a variety of situations where elemental and isotopic analysis is key. We're talking about screening of toxic metals. We're talking about early detection of some diseases associated with the levels of metals, or we are talking about monitoring of chronic patients. And basically what we do is to test new specimens to produce the DBS with a known volume, easy to use for the patient, and then we develop the methodologies for the analysis. If you are interested, we have more information in this website, or you can contact me for further discussion.
The Analytical Biosensor Group is a research group at the Analytical Chemistry Department of the University of Zaragoza and the Institute of Nanoscience and Materials of Acarón. We design a enzymatic nanobiosensor for the determination of chemical compounds in food, clinical or environmental samples. In our approach, the corresponding enzymatic reaction could be monitored by the intrinsic optical properties of the enzyme, mainly absorption or fluorescence, to develop non-invasive methods, or copper-tone indicator reaction, which involves the formation of metallic nanomaterials, mainly nanoparticles or nanoclusters. In this case, the immobilization of the reagents necessary support has allowed us to design test strips in which the concentration of the analyte can be quantified through the RGB coordinates of a picture. We are now mainly focused in this last approach, in which we have designed nanobiosensor for determination of biogenic amines, like putrescine or cadaverine, tyramine or histamine, glucose in drinks, and we are also intending to develop biosensor for determination of aflatoxins in baby and infant formula, tropan alkaloids in cereal, or nitrosamines in baby food. Why is this research line important? Because the final goal of our research line is to develop simpler, quicker, and cheaper system for the determination of the compounds of interest. That is, the design of disposable biosensor or point-of-care devices. Hoping to have attracted your attention to our research line, thank you very much in advance.
I'm here to present you our proposal for the IBRUS. So let me first spend a few seconds in introducing you our research group, the Molecular Oncology Laboratory. We conduct our research at IRB Lleida, but we used to do that at Vall d'Hebron Institute of Research in Barcelona, where we still keep very important collaborations. Currently, the group is composed by 8 people, all very talented and motivated. But as our research is highly translational and really aims to reach patients, we work in close contact with clinicians at the surgery, pathology, and medical oncology units at the hospitals listed here. As you might have noticed, the postdoctoral position in the upper circle is empty, and we are actively recruiting, so the name on there could be yours. Let's move now to the scientific part. We work on precision medicine in colorectal cancer, and this cartoon illustrates the current situation regarding to treatment. We have colorectal cancer patients in one side and approved drugs on the other. And there's nothing telling us which drug is best for each patient. Our aim is to stratify patients to provide a personalized treatment. By doing so, we will reduce unnecessary toxicities and increase the treatment response rates, and that directly impacts on patient survival. We do this for 2 drugs approved for the treatment of colorectal cancer patients in advanced stage, and to stratify patients, we use molecular features such as mRNA, protein, and mutations. The molecular features able to anticipate the response to treatment are named predictive biomarkers. We have identified and confirmed some of them, and the challenge is now to develop assays leading to all the steps required for their clinical approval. And someone has to do that, so would it be you?